Hello, I'm James Keenan from Artensia, and thank you for inviting me to present on our Microbubble drug conjugate and focus ultrasound blood-brain barrier delivery of AAV-2 SIRT-3. So Parkinson's disease is a serious unmet need, and clinical trials with direct surgical infusion of gene therapy and other therapeutics has failed to improve patient outcome. However, Sunnybrook Hospital and other sites have done Parkinson's disease clinical trials demonstrating the safety of focus ultrasound and microbubble BBB opening. And the direct infusion of AAV-SIRT-3 has demonstrated neurorestorative and neuroprotective effects in Parkinsonian rodent models. As well, focus ultrasound BBB drug delivery with microbubble drug conjugates versus the sequential administration of microbubbles and drug increases a drug's dose, perfusion, and retention, avoids systemic effects with unused drug cleared in the liver, and may extend the short in vivo half-life of gene-based molecules. So we developed antibody linkers with binding specificity to AAV2. And we covalently conjugated these to the lipids that form our microbubble shells. These microbubbles were generated, and we added and incubated the AAV2 gene therapy. We first tested this in vitro in a focus ultrasound flow chamber. The microbubble drug conjugates flowed over a cell bed of SY5Y cells, where they were sonicated, and ultrasound imaging used to verify microbubble viability. And the results were that before a focus ultrasound exposure, we have a bubble population. After 600 kPa, the bubbles are destroyed. And we found a significant increase in the transduction in the ultrasound exposed group versus the non-ultrasound group. So we moved on to in vivo. We used MRI to visualize the brain of healthy rodents and to identify targets associated with Parkinson's, the striatum and the substantia nigra. The animals were given an IV injection of microbubble drug conjugates with AAV-2 SIRT-3, and then we used a focus ultrasound LP100 system to sonicate 2 targets in the ST and 1 target in the SN of 1 hemisphere of each animal, therefore using the animal as its own control. Post sonications, MR imaging confirmed the BBB openings in the ST and the SN. So 2 weeks after the procedure, tissue samples were collected and immunofluorescent labeling done so that we could compare the transduction of the sonicated regions of the striatum and the SN versus the non-sonicated. And we found that elevated levels of SIRT-3 were in the sonicated regions versus endogenous in the ST, but not in the SN. So we conclude that it's feasible to develop clinically scalable microbubble drug conjugates with AAV-2 SIRT-3 or other viral gene therapies to treat neurodegenerative disease. As well, the gene therapy retains its ability to transduce after attachment to and destruction of microbubbles. And in vivo, that the temporary permeabilization of the BBB using focus ultrasound and microbubble drug conjugates is a useful technology for non-invasive delivery of AAV-SIRT-3 to brain regions affected by Parkinson's disease. So I'd like to thank the contributors, and a special thanks to the Focus Ultrasound Foundation, as well as the Ontario Research Fund. Thank you.